Yes, level biology 9700 paper 2 variant 1 October November 2025. We will start from question number 5, and question number 5 is figure 5.1 is a photomicrograph of a longitudinal section through part of the stem of the plant. Name one substance transported in T in figure 5.1. In figure 5.1, you can see a spiral helical has been shown over here. It means this is the xylem in which the lignification is in this way. So they have asked about the, so it can be a xylem. So the function of xylem is transport water and mineral salts. And the second function of xylem is to provide support. But here they have asked about only one substance which is transported by this tissue T. So that is, we will write over here, that is water. Water and mineral salts. Now, the second part of this question is state the name of the substance that forms the spiral thickening around the structure labeled T in figure 5.1. And I have told you that this is the lignification. So the process of lignification takes place, which helps the xylem to support the plants. So here we are going to write the name of that substance that forms this spiral thickening. So the lignification takes place by the deposition of the lignin. So I'm going to write the answer over here, the lignin. Lignin. The next question is, a scientist studied the effect of leaf temperature on the rate of transpiration from the leaves of the wheat plant, Triticum aestivum. So the scientist repeated the investigation using the cotton plant, Gossypium hirsutum. The result of the investigation are shown in table 5.1. So the table 5.1 shows the rate of transpiration in both of these species of plants. So you can see that as temperature is from 25 to 45, it means when temperature is increasing, the rate of transpiration in one type of plant, Triticum aestivum, it has been increased. As the rate of temperature is increasing, the rate of transpiration is also increasing in both cases. But if we compare both of these plants, the rate is more faster in one species as comparison to the other. Now the question is, suggest explanations for the relationship between leaf temperature and the rate of transpiration of Triticum aestivum as shown in table 5.1. So why transpiration increases with temperature? First of all, we must know that one, it is increasing. So because we have to explain the relationship between them. So as the temperature rises, water molecules inside the leaf gain more kinetic energy. This causes them to move faster and change state from liquid water to water vapor more rapidly within the spongy mesophyll air spaces. So higher temperature increases the capacity of the air to hold moisture. So this creates a much steeper water potential gradient between the saturated air spaces inside the leaf and the drier air outside. So high concentration of water vapor, lower concentration, stomata through the stomata, so more water will be evaporated. So higher leaf temperature provide the latest heat of vaporization required for water to evaporate from the moist cell walls of the mesophyll cells. So the rate of evaporation is directly linked to thermal energy. The supply of water vapors to the substomatal cavity increases. So I would like to write all these points. Uh, we have to write the minimum 3 and C temperature rises, water molecules gain more kinetic energy. This causes them to move faster and change state high temperature increases the capacity of the air to hold moisture. High concentration of water is inside the leaf and lower outside. So faster diffusion of water vapor out to the stomata. So more water is diffused out due to this high temperature. High temperature, due to high temperature, the most is so higher leaf temperature provide the latest heat of vaporization. So more water is evaporated out. Now, next question. Part B of this. Suggest one difference between the structure of the leaves of T. aestivum and the leaves of G. Hirsutum that cause explain the results shown in 5.1. So here you can see that the difference between these two can be due to the uh, number of stomatas present in both of these. So I would like, because only one mark is there, so G histotum typically has so G histome has sometimes has more stomata has more stomata the lower epidermis on the lower epidermis, whereas whereas the estigum may have more even distribution So this is the one possible answer which we can write. And the second, we can also write the presence of the that the stomatas are protected by trichromes, or we can write one more answer, that is the cuticle. One species may have a thicker waxy cuticle on the upper epidermis. So this is one possible answer. The second possible answer is the presence of a waxy cuticle. One species may have thicker waxy cuticle. Now the next question. Nematodes are microscopic animals that infect a wide range of economically important plant crops, including cotton plants. Nematodes feed on the roots of plants, limiting their growth. When root cells become infected, the nematodes disrupt the plant mitotic cell cycle. This causes the formation of a special type of feeding cell in the plant from which the nematodes absorb nutrients. Cell G is formed as a result of multiple cell cycles without any cytokinesis. Suggest how cell G differ from cell produced during mitotic cell cycles that has not been disrupted by the nematodes. So we are going to difference between these cells. Again, the question is we have to suggest the answer. So we can write that the cell G, normal plant cell has a single nucleus, whereas cell G will contain multiple nuclei because the nucleus has divided several times without the cell itself splitting. So due to which the cell G is likely much larger compared to a normal cell. It has accumulated the mass and organelles of what should have been several individual cells. So the total amount of DNA within the single plasma membrane of cell G is significantly higher than a standard diploid somatic cell because cytokinesis has not been taken place. If the cytokinesis has taken place, that it must be divided into two. As it is not dividing into two, so it has more organelles, its size will be larger, or it has normal, just it is not like the normal cell. So I'm going to write this contain multiple nuclei. Due to multiple divisions of nucleus, its size is also larger than a normal cell. So we can write any one of from any one of these questions. Next question is nematodes can also stimulate a process called endoreplication. This process causes a plant cell to go through multiple S phases during one cell cycle without entering mitosis or undergoing cytokinesis. State how the nucleus of a cell that has been uh, through endoreplication may differ from the nucleus of a cell in the same plant that has not been affected by the nematode. So here I would like to, first of all, the what is S phase? So this is the S phase is that phase in which DNA is replicated. So amount of DNA in the nucleus becomes doubles. So when cytokinesis takes place, then replicated DNA is split into two and the cell divides into two daughter cells. If a cell goes through multiple S phases but no mitosis or cytokine, it is essentially duplicating its genetic material over and over again without ever handling it off to a new cell. So there will be increased DNA, more DNA compared to a normal diploid cell. It will have the larger size. I am going to write the nucleus will contain much more DNA compared to a normal diploid cell. And there will be multiple copies of the genome within single nucleus. Because again, the cytokinosis has not been taken place. So it was question number 4. Now I'm going to start the question number 6. Sorry, it was question number 5, not question number 4. Now, the question number 6 is, alveolar macrophages are phagocytes found in the human gas exchange system. They produce hydrolytic enzymes such as lysosomes to digest pathogens entering the alveolus. State the term used to describe the sequence of nucleotides in the DNA of the alveolar macrophages that code for a protein such as lysosomes. And you know that the nucleotides sequence of nucleotides in the DNA of the alveolar macrophage, uh, such as lysosome, that is the answer is a gene. So the sequence of nucleotides, that is a gene. Very simple question. This is a gene. Second, synthesis of lysosome occurs in 2 stages. The first stage occurs in the nucleus using one strand of DNA to synthesize messenger RNA. State the name of the strand of DNA that is used to synthesize messenger RNA. Very well know that during protein synthesis, one strand of DNA acts as a template strand. So we are going to write its name, which has to be copied by the messenger RNA. So the that is called as the template strand. Name the organelle where the translation of messenger RNA takes place to produce lysosome. Well, again, very easy, the transcription and translation. These are the 2 stages. Translation and transcription takes place in the nucleus. Then the translation takes place on the ribosomes. Ribosomes are attached on the rough endoplasmic reticulum. Ribosomes attached on rough endoplasmic reticulum. So lysosomes destroy bacterial cell by hydrolyzing bonds in peptoglycan. Explain how the hydrolysis of bonds in peptidoglycan leads to the destruction of bacterial cells. So what happens over here? That we have to explain how the hydrolysis of bonds in peptidoglycan will lead to the destruction of bacterial cells. So lysosome play a very important role over here. So what happens? That is the peptidoglycan provides structural integrity and mechanical strength to the bacterial cell wall. So hydrolyzing these bonds break the cross links of the wall, due to which the bacterial cells typically have a much higher internal solute concentration than their surrounding. So without a strong cell wall to resist internal pressure, water enters the bacterium via osmosis. This causes the cell to swell and eventually burst, destroying it. So I have explained, but I briefly I will write the answer. Hydrolyzing these bonds breaks the crosslinks of the wall of pathogens So without a strong cell wall to resist water enters the bacterium enter the bacterium via osmosis. This causes the cell to swell and eventually burst. Next question. Alveolar macrophages are found in contact with sigma-most epithelial cells in the walls of alveoli. Explain how the cells lining the alveoli are adapted for gas exchange. The cells lining the alveoli are sigma-most epithelial cells, and they have a few specific adaptations. These cells are extremely thin and flattened, so this creates a short diffusion distance for oxygen and carbon dioxide to move between the air in the alveoli and the blood in the capillaries. The cell membranes are highly permeable to gas molecules, allowing the rapid exchange. So we have to write these two. Since these cells are extremely thin and flattened, this creates short distance for diffusion. Secondly, the cell membranes are highly permeable, to small gas molecules to exchange easily. And the next question is, describe the role of elastic fibers in the wall of the alveolus. So the elastic fibers primarily, they are made up of protein elastin. They allow the alveoli to stretch and expand as they fill with air during inhalation, which increases the surface area for gas exchange. They allow the alveoli. You must know the characteristics of these types of fibers. Stretch and expand, which increases surface area for gas exchange. They also provide they also provide elastic recoil, helping to push air back out of the lungs. So it was all about the question number 5 and 6. Please like, share, and subscribe my channel. Thanks for watching.